Just testing the audio there. All right. Uh, well, there's like a minute delay. I hope everybody's doing well. Thanks for tuning in. We're going to pick back up with the cardiovascular unit. We've got a couple more slides on the heart. Going to talk about some cool physio stuff today, physiology stuff today. And then we'll move into the beginning of the vasculature. Then we'll finish up the vasculature on Thursday, I expect. And here we go. Okay. All right. So that's where we left off with the artificial heart. So the cardiac cycle. The cardiac cycle it involves all the events of the heart, and they're both physical and electrical, and we'll talk about both parts. So during a cardiac cycle, the pressure in the heart chambers rises and falls, right? When we contract the heart, when the heart muscle contracts, that's going to build pressure in the chambers of the heart. And these pressure changes are going to open and close the valves of the heart. So during atrial systole, this is atrial contraction, and ventricular diastole, that's ventricular relaxation, the ventricles will be relaxed. All right, the AV valves will be open and the semilunar valves will be closed. Now remember, the AV valves are between the atria and the ventricles. And then the semilunar valves are the valves between the ventricles and the large arteries leaving the heart. So remember, the atrioventricular valves include the tricuspid on the right side and the bicuspid or mitral valve on the left side. The semilunar valves include the pulmonary valve on the right side, which leads to the pulmonary trunk and then pulmonary arteries. And the aortic valve is going to lead from the left ventricle to the aorta, the largest artery. All right, so about 70% of the blood flow flows passively from the atria to the ventricles, as we said before. Basically, the ventricles relax, and about 70% of the blood that's in the atria is just going to fall into the heart. Um, and again, the AV valves are open. Those are the tricuspid and the bicuspid. They're open. Now we are going to contract the atria, which pushes the remaining 30% of blood that's in the atria into the ventricles, and that's called the atrial kick. We basically kick out the remaining 30%. So as I mentioned before, atrial fibrillation, which is a shaking of the atria, a rhythmic shaking of the atria, that's not typically fatal, at least not right away, but you do need to correct it because it can lead to complications. It can allow blood to become stagnant in the heart, which promotes blood clot formation, and traveling blood clots are bad things, seriously bad things. They cause heart attacks and strokes. All right. Anyway, moving on. During ventricular systole, that's when the ventricles are contracting, and atrial diastole, that's when the atria are relaxing, the AV valves are going to be closed. And so basically what happens, if I can go back to a picture of the heart. Okay, here we go. When the ventricles contract, they're going to squeeze, and when they squeeze, they're generating pressure. You could say they're putting pressure on the blood that's already in the ventricle. And as we increase the pressure in the ventricle, that's going to push back against this AV valve, the tricuspid valve here on the right side. Hold on, I've got to sneeze. Sorry. All right, sorry about that. So as we generate pressure in the right side, we're going to uh, push against this valve and basically push it shut. Pressure builds in the ventricle, and the tricuspid valve will collapse. Same on the left side. Pressure builds up in the left ventricle, the bicuspid or mitral valve will shut. And what this does is when this valve shuts, it prevents blood from pushing back into the atrium. Prevents blood from pushing back into the atrium. All of these valves, in short, prevent the backflow of blood. Now, the right ventricle, as we continue to contract, we're going to continue to build up pressure in the ventricle. Same on the left side, but I'll just talk about the right for now. We're going to continue to build pressure, and once the pressure in the ventricle is higher than the pressure in the pulmonary trunk, then the pulmonary valve will open up and blood will flow out of the right ventricle into the pulmonary trunk. All right. And then as pressure drops in the pulmonary trunk, okay, as the right ventricle relaxes and pressure drops, we're going to see the closure of this valve, which again prevents the backflow of blood into the right ventricle. If we allowed blood to flow back, our heart would be less efficient, right? We'd be essentially pumping less blood per heartbeat, okay? All right, so to go back, the chordae tendineae, some textbooks will call them the chordinae tendineae, but either way, they prevent the cusps of the valves from bulging too far backward into the atria. And the chordae tendineae are these tiny little cords here. Okay, you see these finger-like structures, these little finger-like structures sticking out of the bottom of the ventricle. Well, those are the papillary muscles. Okay, these are the papillary muscles, and attached to them are the chordae tendineae, which are attached to the AV valves. And the AV valves, remember, prevent that backflow of blood, and the chordae tendineae, attached to both the valve and the papillary muscle, prevent the valve from bulging too much when uh, pressure increases in the ventricle. All right, so when the ventricles are contracting, or in other words, during ventricular systole, the atria will be relaxing. Okay, it's not perfect on off between the two. They don't perfectly switch off. Like one contracts, the other relaxes in the at the exact same moment. But in general, just in general, when the ventricles are relaxing, the atria will contract, and when the ventricles are contracting, the atria will relax, and blood will flow into the atria from the vena cava and the pulmonary veins. Okay, so to go back to the heart, here we see the vena cava. Okay, that's the plural. There's a superior vena cava, and right here there's an inferior vena cava. And these are the largest veins. And again, they return blood to the right atrium along with the coronary sinus. All right, so while the ventricles are contracting and generating force and preparing to shoot blood out downstream, the atria will be relaxing, and that allows them to fill. Okay, we're going to fill the atria with blood while the ventricles are relaxing, so that when the ventricles are done contracting, so that while the ventricles are contracting, so that once they're done contracting, we can then let this blood flow into the ventricles and fill them again. All right. So we mentioned ventricular pressure, it increases, and that's going to eventually open the semilunar valves, all right, the pulmonary and the aortic valves. Hey, Fauziat and Kadra, I'm just looking up now. If anybody's got any questions, feel free to just shoot them up there. I'll take a look over periodically and answer them. All right, so the blood flows then into the pulmonary trunk and the aorta once pressure in the ventricles is great enough to overcome the pressure in those arteries, those large arteries. All right, so a heart beat through a stethoscope sounds like lub dup. All right, so when the heart beats, you put the stethoscope up to the auscultatory areas. Those are the areas where you can hear the heartbeat. It sounds like lubbed up, lubbed up, lubbed up, lubbed up, lubbed up, lubbed up. And the sounds are due to the closing of the heart valves, and then that's going to generate vibrations. And what you're hearing are, of course, are the vibrations associated with the sudden slowing of blood during contraction and relaxation of the chambers. So the lub sound is the first heart sound. It's the lub in lubbed up, and it occurs during ventricular systole, and it's associated with the closure of the AV valves. Dup is the second heart sound, which occurs during ventricular diastole or relaxation. All right, and it's associated with the closing of the pulmonary and the aortic semilunar valves. A murmur is an abnormal heart sound, which is derived from incomplete closure of the cusps of a valve. The cusps of the valve, let's see if we have a good picture. Oh, I know we have one back here. Yeah, here you can see the cusps of the valve. Okay, here also with the AV valves. Okay, easy to see here with the tricuspid, you got 1, 2, 3. Bicuspid, you got 1, 2. All right. So the murmur is due to incomplete closure of the cusps of the valve. All right. And this can be due to stenosis, which is a narrowing, a narrow opening in the valves, or calcification of the valves, or any number of things. All right, here are the auscultatory areas. That's a fun word. I'm actually going to insert a text box and type that out. Text box. There we go. Auscultatory areas means places where you can listen. Oh, do not tell me this froze. Okay. Good, good. It's back. All right. So here you can see the pulmonary area, okay, which is below the second rib on the left side of the chest. And this is where you can see in the heart there's the pulmonary trunk. And here you're going to be listening for the pulmonary valve. Over here on the right side, you see here's the sternum in the middle. So over here on the right side, also below the second rib, you can hear the aortic area. It's where you'll be listening for the aorta, aortic valve, that is. All right. So down here below the fourth rib, we've got the tricuspid area. It's over here on the left side near the sternum. And that's where you're going to hear the tricuspid valve. Okay. And further left, down here toward the apex of the heart, below the fifth rib, we have the mitral area, where again you'll be listening to the mitral valve. So cardiac muscle, these cells form branching networks. They also have intercalated discs between the cells which contain gap junctions so that action potentials can be spread through the network of cells. And actually, I don't think we have a good picture of this. So why don't we look for one on Google? Cardiac muscle. Okay, so here's a microscope slide. You can see here those intercalated discs between the cells. Remember that skeletal muscle fibers are very long. Skeletal muscle fibers run the length of the anatomical muscle, and in the case of the sartorius, that can be like up to 2 feet long. So that those cells are very long. In the heart, cardiac muscle cells are short. Uh, they're still bigger than most cells, but they're short as far as muscle goes. And so you can see from here to here, from one intercalated disc to the next is a muscle cell. All right. Cardiac muscle also is usually uninucleated, meaning has usually one nucleus, but it can have like two or three. Uh, but it's not like skeletal muscle where there were tons of nuclei. Just usually one, maybe two or three, depending on the cell. All right, now let's see if we can get a better. They have a cartoon picture? Yeah, here's great. Here's great. All right, so you can see that these cells also branch. All right, they branch. All right, and they've got this is striation. They also have striation like the skeletal muscle cells. This cartoon's not showing the intercalated discs. Hmm. Great. Here's a good one. You can see the intercalated discs. You can see the branching cardiomyocytes. Okay. All right. So why are the intercalated discs important? Well, one, they hold the muscle cells together, but two, they have gap junctions. All right, embedded in the intercalated disc. If you don't remember, a gap junction is basically an opening from one cell into the next. Two cells are connected to each other, and there's some like open windows between them. It's kind of like if you had a duplex house, right? It's where you take a house and then you put up a wall down the middle, so now you got two homes in there, right? Now imagine there's a window between the two sides of the duplex. That's kind of like a gap junction. Here you can see in the intercalated discs, we have some of these gap junctions here, which are basically open windows between the cells. And so we talked extensively in ANP1 about action potentials, how they're generated, how they're propagated. But all right, so in the skeletal muscle, the sodium, just like the cardiac muscle, the sodium floods in, okay, which changes the charge across the membrane, which opens up the potassium channels and lets the potassium flood out. And now we've depolarized our membrane. Now, an adjacent region of the membrane down here will begin to get depolarized. All right, the change in voltage down here is going to cause sodium channels down here to let sodium flood in. The change in voltage there lets potassium flood out. Okay, and now we've depolarized this region of the membrane. And then because there are gap junctions between this cell and the next cell, which would be over here, uh, we're going to see that action potential continue as sodium moves from inside this cell into the next cell, which is going to open up the potassium channels and let the potassium out. All right, so the cardiac muscle fibers, or cardiomyocytes, form a functional syncytium. That's a hard word to say. Functional syncytium, which is a mass of cells that function as a unit. What does this mean? Well, basically, the heart functions as a unit, it contracts as a unit. Now, not all of the heart contracts at the same time. Different regions of the heart contract at different times, but we're going to see contraction begin in one area and then spread to the rest of the heart. So the atrial walls are called the atrial syncytium. They will contract as one unit, and then the signal to contract will be propagated to the ventricles, where the ventricles will contract as a unit, and we'll see that's called the ventricular syncytium. So the cardiac conduction system is a group of clumps and strands of specialized cardiac muscle tissue, which initiates and distributes impulses throughout the myocardium. So what does this mean? With the cardiac conduction system, we're going to be talking about the structures in the heart that initiate or begin the signal to contract and then spread it to the appropriate parts of the heart at the appropriate time. So in ANP1, we mentioned several times that The nervous system does not tell the heart to beat. The brain does not tell the heart to beat. All right. If you were to be decapitated, the heart would continue to pump. If you take a heart out of someone's chest, I feel like I got to sneeze again. Sorry. If you take a heart out of someone's chest, it will continue to beat. And you can actually, as long as you continue to supply the heart outside the body with blood that is rich in oxygen and nutrients, the heart will continue to beat outside the body for a long time. All right, and why is this? Because the heart tells itself to beat. The heart it initiates the signal to contract itself. And it all begins with a clump of cells that make up the SA node. And then the SA node, when it depolarizes, that action potential is spread through the atrial syncytium so that the atria contract as a unit. And then there are junctional fibers between the atria and the ventricles, and they're going to continue the signal, propagate the signal down to the AV node or the atrioventricular node. This is another node, as you might guess, found between the atria and the ventricles. The signal is then conducted through the AV bundle, the bundle branches, the Purkinje fibers, or a lot of people say Purkinje. I hate that, but a lot of people say Purkinje. And then down to the ventricular syncytium. So why don't we go ahead and take a look at that? All right, why don't we go ahead and take a look at that? Up here is the SA node. Okay, the SA node is near the superior vena cava. Okay, it's up here. And so the signal to contract begins here, and that's going to be spread through fibers through the wall of the atria. And so the atria contract as a unit first. Okay, so we see atrial systole, contraction, and then that signal is propagated after the atria contract down to the AV node or atrioventricular node. Okay, and again, this is the node between the atria and the ventricles. The signal is then carried by the AV node down to the AV bundle, which is this short stretch here between the AV node and the bundle branches. Okay, you see the bundle branches are these large branches that pass through the center wall of the heart. Center wall of the heart, by the way, is known as the ventricular septum. Sorry, it took me a second there. I don't know why. Ventricular septum. So the bundle branches pass through the ventricular or interventricular septum, the septum or wall between the ventricles. All right, and then from there, the bundle branches come down toward the apex and then curve back up the wall of the heart. And then branching off of the bundle branches are the Purkinje or Purkinje fibers, okay, which are going to continue the signal, all right, out to the cardiomyocytes. Now, the cardiomyocytes, okay, they form kind of a ribbon-like sheet that wraps around the heart, okay? It's maybe a little hard to see under normal circumstances. It just looks like a solid wall of muscle, right? But in reality, there are layers, this kind of ribbon-like pattern which envelops and becomes the heart here. So that when the ventricles contract, they contract in a sort of twisting motion. All right. Let's go back, see if I missed anything. The, the SA nodes, these are the pacemakers. So they initiate the rhythmic contractions of the heart. The internodal atrial muscle, this is atrial muscle between the two nodes, the SA and the AV nodes. They conduct the impulses from the SA node to the atria. The junctional fibers conduct impulses from SA node to AV node, from the sinoatrial node to the atrioventricular node. Then the AV node conducts impulses to the AV bundle, as we said. It also delays the impulse so that the ventricles don't contract while the atria are contracting. If the ventricles in the atria contracted at the same time, this would prevent ventricular filling. This would prevent the ventricles from getting full of blood before they contract. All right, and so if they're not getting filled with blood when they contract, then the ventricles aren't being as efficient as they need to be. All right, so the AV or atrioventricular bundle, also called the bundle of His, it conducts impulses between the AV node and the bundle branches. It's that short little bit we saw right here. Okay, kind of embedded in the wall there. All right, and then from there we have the left and right bundle branches that split off from the atrioventricular bundle, and they conduct their impulses down to the Purkinje or Purkinje fibers, which are large fibers that conduct the impulses to the myocardium or heart muscle. Great. All right, so again, you can see that here the SA node conducts impulses through the atria. All right, and then the impulses are conducted down to the AV node, and from the AV node down through the bundle of His or AV bundle to the right and left bundle branches, which come down toward the apex and then curve back up. Okay, and then off of the bundle branches, we see the Purkinje or Purkinje fibers. One more thing. Can't remember if we talk about it or not in here. I don't think it's in the slides, so I'll go ahead and talk about it. Interesting thing, if you have a problem with the SA node, okay, the AV node can actually take over. So if for some reason the SA node is damaged, and this does sometimes happen, the AV node can take over, and the AV node will become the pacemaker. Normally, it's the SA node that generates that signal to contract, and so the SA node is called the pacemaker of the heart. If it's damaged, the AV node can take over. Now, the AV node, if it does take over, it depolarizes more slowly than the SA node does. All right, and so if you have damage to the SA node, when the AV node takes over, you will see the heart rate drop. The heart rate will drop. If there's damage to the AV node, the bundle of His can take over, okay, and it can actually depolarize as well. And then all of the parts of the cardiac conduction system, I suppose, can depolarize, but once you get down to the bundle branches, let me take a step back. At each step, the heart rate gets slower and slower. So once you get down to the bundle branches, the heart rate is slow enough that you won't live. All right, so you'll never see the Purkinje fibers take over because the heart wouldn't contract often enough for you to pump blood through the body and keep all your tissues alive, especially the brain. So that was a lot. Just in short, SA node is the pacemaker. If it's damaged, the AV node can take over, but you'll see the heart rate drop because it depolarizes more slowly. We don't have bonus questions on our exams in here, but if we did, this is just one of those kind of fun little science trivia things I'd point out that might be a bonus question. The AV node depolarizes more slowly than the SA node. By the way, these are both constantly depolarizing on their own. They have leak channels in their membrane, which causes them to slowly let sodium back in and then eventually depolarize, let potassium out. They're both constantly in the process of depolarizing. The SA node depolarizes more quickly and the AV node depolarizes more slowly. The AV node depolarizes more slowly so that it can allow the SA node to be the pacemaker. If the AV node depolarized more quickly, it might take over, it might take control of the system, kind of ignoring the input of the SA node, and then you would have chaos with different parts of the heart contracting possibly at the same time. All right, so moving on, we've got the electrocardiogram, or ECG, or EKG. By the way, why ECG, EKG? Another little trivia question. The word cardio is Greek, and in the Greek alphabet, they don't have a letter C, just K. And so the word cardio in Greek is spelled Kardio. So that's why there's EKG. ECG is the exact same thing, just using the English spelling with C for cardio. All right, so the ECG is a recording of the electrical changes that occur in the myocardium, or the heart muscle, during the cardiac cycle. And it's used to assess the heart's ability to conduct impulses. So there are deflections in the normal ECG or EKG wave. Uh, and we should be familiar with the P wave, the QRS complex, and the T wave, and we will see these in a second. But the P wave, all right, is atrial depolarization. When we see the P wave, we're seeing atrial depolarization. And this occurs just prior to, just before atrial contraction. All right. And why is that? It's because what we're detecting are changes in voltage, and we're detecting it across the chest wall, right? We're detecting our ECG from outside the body. And again, what we're detecting is the electrical activity of the heart. So the impulse travels through the atria, and then the atrial muscle then begins to depolarize and contract. All right, the electrical signal goes first. It's much faster. That's followed immediately by contraction. The QRS complex has three waves labeled Q, R, and S, and they represent ventricular depolarization. And this again is going to occur just prior to, right before, ventricular contraction. Uh, that's followed by the T wave, okay, which represents ventricular repolarization, and that's where the ventricles repolarize. And this occurs right before ventricular relaxation. Now, you may be asking, where is atrial repolarization? And that's hidden in the QRS complex. The QRS complex is a much bigger electrical event, and so we aren't able to detect atrial repolarization because it's happening during the QRS complex. And so here you can see the heart. In green, you see depolarization. I'm sorry, not green. Yellow, you see depolarization. And then green will actually represent repolarization. So let's go through these vertically. All right. So the first one up, we see the atria begin to depolarize. Of course, you know right up here is where we would find the SA node. This is where it begins. We see the atria depolarized. That is propagated through the right atrium and then over to the left atrium. And that gives us this little deflection right here. All right. Once the atria are fully depolarized, we have completed our P wave. And so you can see here the wave begins, it crests, and then it finishes as depolarization spreads through the atria, or as the atria depolarize fully, we see the electrical activity of the P wave. Now, moving on, we have the QRS complex, and you can see there's QRS right here. And the QRS complex represents ventricular depolarization. All right, and so the signal has been propagated through the AV node, the bundle of His, the bundle branches, okay? And we're also going to see the atria repolarizing here. Now, we can't see that on the recording of the electrical activity. We can't see that on the ECG because the QRS complex, the propagation of that signal through the ventricles is just a much larger electrical event, and so it masks atrial repolarization. All right, now moving on, we can see that the QRS complex has completed and the ventricles are fully depolarized. Now the ventricles will begin to repolarize again. We see the beginning of the T wave or T deflection, okay? And then as the ventricles repolarize, are fully repolarized, we'll see the T wave completed, okay? All right, so this diagram is a very famous one, actually. We had to learn it at the med school. It used to be physicians would carry it around, a little laminated copy in their coat pocket so they could check it when they needed to. And this has pretty much all of the information, minute by minute, second by second, that you might need to know about the cardiac cycle. All right, so up here you can see the first line shows you atrial systole, atrial diastole, atrial systole, atrial diastole. All right, and so down here we have ventricular systole and diastole. And you can see when atrial diastole begins, ventricular systole begins. All right. Now, a little bit later, ventricular diastole begins, and we have this period where both the atria and the ventricles are beginning to relax. Then atrial systole occurs while the ventricles are still relaxing. Okay. Moving on down here, you can see the recordings of pressure. Okay. Uh, so pressure in the heart. Let's see here. Where are the colors? Where's the color scheme? I don't see it, but okay. They label it here. We have the aortic pressure is this blue line up here. This is pressure in the aorta. And so pressure in the aorta builds after ventricular systole has begun. Okay, it's falling until then, and then it builds shortly after the ventricles begin to contract. Okay, when the AV valve closes right at the beginning of ventricular systole, we're going to have this period of time where we're just building up pressure, this very small period of time where we're just building up pressure. And then the aortic semilunar valve will open and pressure in the aorta will shoot up. And then it'll begin to drop off. Okay, and as pressure in the aorta drops off, we're going to see closure of the aortic semilunar valve. Okay, and then during ventricular diastole or relaxation, pressure in the aorta will slowly drop as the blood that's in the aorta moves on to the tissues. Okay, continue to relax the ventricles. We see the aortic blood pressure continue to drop until eventually we contract the ventricles again. Then we're going to see the the opening of the aortic valve. Pressure in the aorta shoots up again and then comes back down. We see closure of the valve and then pressure drops. And anyway, that was a long-winded explanation of how you could use this diagram. It's very useful. If you want to go into emergency medicine, I would definitely become very familiar with it. Cardiologists need to have it memorized. All right. You can also see down here ventricular volume. So during ventricular diastole, during ventricular relaxation, you're going to see the volume in the ventricles increase. Okay. Then during ventricular systole, we're going to see the volume drop. In fact, right at the same moment where the aortic valve opens, the volume in the ventricle is going to drop. Why is that? Because the blood from the ventricle moves into the aorta. So the volume of the ventricle drops. Then during ventricular diastole or ventricular relaxation, we see passive filling of the ventricles. This is about 70% of its volume here is passive filling. And then right here at this moment where we see atrial systole begin, that's atrial contraction. We get that atrial kick where we get the last 30% of the blood kicked into the ventricles. All right, now we have ventricular systole again. We're going to contract, build pressure until we open the aortic valve, and then volume will drop in the ventricles, particularly the left ventricle, since we're talking about the aortic. All right, good. Here's the ECG. So you can coordinate, or you can correlate all of these events to the ECG as well. So here you have that P wave, and that's the P deflection, the depolarization of the atria. The depolarization of the atria is immediately followed by atrial systole. And so you can see here, the P wave begins pretty much on the left side of this graph, okay, and it's completed over here, move up. All right, atrial systole begins shortly before the end of the P wave. All right. So we're just pointing out here that again, the events of the ECG are followed shortly after by contraction. They don't happen at the same time. It's followed shortly after. So in the middle of the P wave, atrial contraction, atrial systole begins. We have the signal passing here from the SA node through the atria. Okay, and then a few fractions of a second later, the atria will begin to contract. Moving on, we see the electrical signal passing through the ventricles. Okay, that's the QRS complex. The ventricles are depolarizing. And in the middle of the QRS complex, right around R, that's when ventricular systole begins. Okay, and so the signal's already started to pass through the ventricles. The signal's passed about halfway through the ventricles before the ventricles even begin to contract. Then T down here, we see ventricular repolarization. That's relaxation, well, followed by relaxation of the ventricles right here with ventricular diastole. All right, and then finally you can coordinate that with the heart sounds. And so lub, that's the first sound, and that is associated with the AV valves closing, which you can see occurs right here. And then dup, okay, is associated with the semilunar valves closing, which is at the end of ventricular systole. Okay. Lub's at the beginning of ventricular systole. Dup is right after it ends. It's a fun little diagram. If you do manage to memorize this, I mean, kudos for real. I mean, you might be ready for med school if you can get this memorized. All right, so regulating the cardiac cycle. So the SA node normally controls the heart rate. And then the sympathetic and the parasympathetic fibers are going to modify the heart rate in response to changing conditions, such as physical exercise, body temperature, fight or flight responses, and then the concentration of various ions like obviously potassium and calcium. Now, as we said, just again, SA node is the pacemaker of the cell. The SA node is the pacemaker of the heart. It controls contraction in the heart, controls the heart rate. Brain does not tell the heart to beat. However, it does tell the heart speed up or slow down. Okay, it does tell the heart speed up or slow down. So if you are under the effects of certain drugs, for instance, epinephrine, which is you get an injection with the EpiPen. That's going to speed up your heart rate, okay, because it mimics the activity of the sympathetic nervous system. The sympathetic nervous system, in other words, is going to, let's forget the EpiPen example. That was a bad example. The sympathetic nervous system is going to tell the heart rate speed up. You saw a saber-toothed tiger, it's going to tell the heart rate speed up. We need more blood, more oxygen, more nutrients spread throughout the body, speed up. And if you see somebody you really like, you got feelings for, your heart rate's going to speed up. Same fight or flight response. Okay, that's the sympathetic nervous system. The parasympathetic nervous system is going to tell the heart rate going to tell the cardiac conduction system to slow down, slow the heart rate. Okay, and so the parasympathetic nervous system innervates the heart through the vagus nerve. Okay, and then the sympathetic nervous system, well, not the vagus nerve. It actually has branches off of the spinal cord. We talked about that last semester. We also have baroreceptor reflexes, and the word baros is Greek for pressure. So baroreceptors detect pressure. We have them in the carotid sinus. We also have them in the aorta, and they detect your blood pressure. And if your blood pressure is too high, they're going to tell the heart to slow down to decrease the pressure in the vasculature. And if your blood pressure is too low, they're going to tell the heart to speed up and increase pressure in the vasculature. In fact, there's this baroreceptor reflex in the carotid sinus. Basically, you know that feeling when you stand up too quickly and you feel lightheaded? That's because you stood up too quickly and blood left essentially um, the arteries, and mm, due to the change in your position and gravity, blood pressure drops in the carotid sinus. We'll say that. That initiates a reflex where you are going to tell the heart to speed up and increase the blood pressure that way. I'm maybe not being clear about this. That's how it works normally. Young, healthy people typically don't get that feeling because the baroreceptor reflex works. It tells the heart to increase blood pressure as we're standing, and then we'll avoid that feeling. Every once in a while, young people will manage to trigger the response, or fail to trigger the response, and then get that lightheaded feeling. Older people frequently get that feeling as the baroreceptor reflex begins to fail. That's what I was getting at. I know this is very, very confusing stuff. All right, so the cardiac control center in the brainstem regulates the autonomic impulses of the heart. Okay, and so you can see here cardiac control centers in the medulla oblongata. Okay. Uh, yes, in the medulla. I wanted to, yeah, get that straight in my head real quick. So here's the reflex. You got the receptors, the baroreceptors, which detect changes in blood pressure. Now, these baroreceptors are embedded in the walls of blood vessels, and so that's how they're able to detect blood pressure. And through afferent or sensory neurons, they will convey that message to the central nervous system, particularly in the spinal cord and the uh, medulla oblongata here. And then from there, we will see a response sent out through efferent fibers to an effector. In this case, the effector is the heart. Okay. And so you can see up here in the carotid sinus, also in the aorta, there are uh, baroreceptors embedded in the walls of these vessels. And they detect changes in the pressure, which will be conveyed over to nuclei in the medulla oblongata. Now, the medulla oblongata and the hypothalamus integrate this information as well as the cerebral cortex. And we integrate all of the incoming information and then we generate a response which is sent out in this diagram over the vagus nerve or down here out of the sympathetic trunk if it's a sympathetic response to the heart where we tell the SA or the AV nodes to speed up or slow down the rate of contraction. Speeding up the heart rate is going to increase blood pressure, and relaxing the heart, that is slowing down the heart rate, is going to lower blood pressure. So for arrhythmias, these are altered heart rhythms, and there are several types. Fibrillation is an uncoordinated, chaotic contraction of small areas of the myocardium or the heart muscle. Atrial fibrillation is not life threatening, as we've said before. However, ventricular fibrillation is often fatal. And the reason for that is we need the ventricles to pump that blood out, either to the lungs or to the tissues. We need to send that out on its way. On the systemic circuit, we need to send that blood a very long way, all the way from the heart down to my big toe. It's got to get all the way there. Maybe not on one heartbeat, but you know what I'm saying. We got to pump blood a long ways in the systemic circuit. And if the ventricles are not fully relaxing and contracting at the right times, then we're not going to be able to fill the ventricles up fully with blood, and then we're not going to generate enough pressure to send blood all the way down to the little big toe. All right, so tachycardia is an abnormally fast heartbeat, usually over 100 beats per minute while the person's at rest. Now, if you're exercising, over 100 beats per minute's fine. Bradycardia is an abnormally slow heartbeat below 60 minutes, 60 beats per minute at rest. Although people who are physically fit, and I don't mean they're in shape, but they sit around and watch a lot of TV. I mean people who go to the gym and really work out. And long distance runners, especially people who do cardio exercise. Uh, it's not unusual for them to have a resting heart rate below 60 beats per minute, and you may actually hear them brag about it sometimes. All right, that character Chris on Parks and Recreation, you know that TV show Parks and Recreation, super fit guy played by Rob Lowe. I think I saw in one episode he was bragging he had a heart rate of 28 beats per minute, resting heart rate of 28 beats per minute, which is like crazy low. That would kill most people. The reason, by the way, they're able to have sustain a heart rate so low is because through cardio exercise, their heart has become more efficient. Doesn't need to pump as often. All right, so flutter. This is a rapid, regular contraction of a heart chamber, usually around 250 to 350 beats per minute. A premature beat is a beat that occurs before it's expected in the normal cardiac cycle. And this often originates from a condition called ectopic pacemaker. In other words, there are parts of the cardiac conduction system that have gone rogue and are generating their own signals, uh, isolated from the rest of the conduction system. All right, so the artificial pacemaker we are probably familiar with. Generally, people just call them pacemakers. It's a device used to treat disorders of the cardiac conduction system. In other words, it's a device used to treat arrhythmias. It can be implanted in the body. The old ones were external, they were outside the chest that had a little wire that went down into the heart. Um, they are battery powered and they will uh, keep the parts of the heart conducting in rhythm. All right, so we've actually been talking now for almost an hour, and I want to give you guys a chance to ask any questions. If you have any questions, please shoot them up on the board. I'm going to wait here a minute, and I think we'll actually tackle the vasculature on Thursday. This was a lot of information. Only about 10 or so slides, but a lot of information today. Okay, well, I guess we'll call it quits for today. Do send me any questions you've got through email. I get people don't want to ask in front of other people. Maybe they're a little embarrassed or something. But yeah, I am not gonna make fun of you. Just send me any question, no matter how ridiculous. I am happy to answer it through the email. Okay. All right. Well, thank you guys, and yeah, we'll see you Thursday.